So hello everybody, I'm Sybil from the University of Nottingham and I'm working with Dr. Sharp. So as you can see, I'm a cell biologist and I work on a gene that can help to prevent cancer. And this gene produces a protein called LIMD1. Previous studies have shown that LIMD1, when it's lost in mice, these mice without LIMD1 will develop lethal lung cancers. So what we want to know is what happens or what goes wrong in the body when LIMD1 is missing. Firstly, however, we have to work out what LIMD1 normally does. So to do this, my colleague captured this video of a live human cell to show how LIMD1 behaves within a human cell. LIMD1 here is in green, the very bright green spots. And in red, we have a clump of proteins, which we call the microRNA-induced silencing complex. So it's just a very large clump of proteins that work in the cell. And if we look at these three spots in particular, you can see the red and the two green. If you watch it, you can see that the green clumps of LIMD1 slowly move towards the red clumps, and then after a while, it comes off again. So what we believe is that LIMD1 actually helps to control gene expression in the cell by interacting with this miRISC. To give you a crash course, we have a very long genome. And in order to express a gene, you have to take that gene, the DNA that spells out that gene in the genome, and copy it into a much shorter piece of RNA. This RNA then has different specific proteins binding to the ends of it and onto the region that encodes the gene on it in order for production of protein to occur. So that's the normal gene expression. When we have microRNAs, which are very tiny little RNA strands, this can stop protein production from occurring by binding onto a protein that's called AGO. So this AGO then recognizes the protein-producing RNA, and that recruits the rest of the miRISC, forming that red blob at the end, and that stops any further protein production. Our hypothesis at the moment is that the AGO binds to that EIF4E, which is a protein binding to one end of the protein-producing RNA. And what we think is that LIMD1, our protein of interest, is actually acting as a scaffold to hold these two proteins together. To test this theory, I attached AGO, the AGO protein, to an artificial surface, and then I added EIF4E protein to see if it would stick. And from this Western blot, you can see that EIF4E sticks to AGOs 1 and 2. When I added LIMD1 onto this experiment, I was moreover able to see that LIMD1 attached to both EIF4E and AGO at the same time. This supports our current hypothesis. To finish off my thesis, I'm going to look at where exactly the contact points are so that I can have a very exact map of what is happening in the cell. And hopefully in the future, I'll be able to find out what the 3D structure of LIMD1 is so I can complete this hypothesis of how LIMD1 works as part of the miRISC to prevent cancer. That's it for me.